Everyone, my name is Francisco García. I'm a forestry engineer working at the Higher Technical School of Agricultural Engineering in the University of Castilla-La Mancha. We are in Albacete, in Spain, and my research team is formed by Manuel Andrés, Consolación Pig Baena, Francisco López Serrano, Francisco Antonio García Morote, Marta Isabel Picazo, and Eva Rubio. We are being helped also by the Department of Soil and Water Conservation, PEVAS, the SIC, which is in Murcia, in Spain also. Today I'm going to talk about our research for an index for a quantitative analysis of the soil quality of Mediterranean forest. So to begin with, I'll start with this introduction in which I must tell you that here in Spain is quite common in our forestry areas to have them treat in one way or another using silvicultural treatments or mostly vegetation thinning. As you can see in the images, most of them are doing by using mechanical tools or even using fire to clean shrub areas or to eliminate vegetation remnants, mainly. In the last years, those treatments have been risen up, and obviously, if you think and you cause an impact in something that is connected to the soil, then it's most likely of the soil to be altered in one way or another. When these changes happen, the balance between the psychochemical, the microbiological, and the biochemical properties can change a bit. And how can we know the quality of the soil? Well, we have a wide range or a wide variety of edaphic properties that we can choose from. We can study the pH, we can study the moisture, etc. But if we study all of these parameters individually, we are just only getting a piece of the cake. We don't have the whole information. So to solve this problem, to solve this issue, some researchers in the last few years, as for example Bastida or Zornoza, have proposed to integrate all of these parameters, all of these variables, into multivariate indices to get all the information of the changes that can happen in the soil. And that's have been our main reasons of why we have developed this soil quality index. And how can we define a soil quality index? So we can say that this index is like a group of data, a minimum set of data, physical, chemical, and biological variables that when we use together, when we use them together, they can give us or they can provide us with a reliable numerical data about soil functionality in a given area. And how can we know how much a soil has been altered? You have to compare a soil which is disturbed with a soil which is undisturbed. But here we find a little problem. Human activity has changed almost every piece and every place in the world. So when you arrive at a place that has not been altered, you cannot say that it's 100% natural. So the best soils that we have found and we have used in our research as control soils are those which are not being disturbed for at least a period of 30 or at least 30 or 40 years. And that's the control soils that we have used in our research. What's our main objective? So we want to develop with this investigation a multivariable soil quality index. Is everything okay? I hear some noises. Okay, and then I'm going to continue. So as I was saying, our main objective was to develop a multivariable soil quality index, which can be applicable to the Mediterranean forest of the Cuenca Mountains range. What are the main advantages of this index? That I am hearing something or someone speaking. Is there any question or any problem? Or can I
just muted them. They can now go ahead, please.
Okay. So I was telling you that the main advantages we have found is that using the index, in the index is we use an adequate number of physical, chemical, and biological variables of the soil to correlation it with. It is also easy to measure for a great variety of users and field conditions. And also the index have a great response to changes. And that's because we have included in the index the microbiological parameters that can react with relative speed to changes in the soil. Okay. The main disadvantage that we have found in our investigation is that when we got all the data in the lab and you work with them at these first stages, we end with a ton of data, with a big amount of data, and we have to handle and determine this. So it can be at first a little bit challenging, but over that we haven't found any more problems in this way. So here in this slide, I'll show you the study areas that we have used. All of them are located in the Cuenca Mountains range in the province of Cuenca in Spain, which is in central eastern Spain. And as you can see, we have chosen 5 places, okay? But we've got 7 sampling sites. This is important for later. Those places, those areas, here in Spain have a particular name, but just you to know, there are like forestry type areas with a special kind of management and protective state, and you can use them for many purposes. You can go there sightseeing. You can even go, if you have the permission, you can extract wood, etc. The main vegetation that we have found there is mainly with the Pinus nigra subspecies salmani, which is the black Spanish pine forest, which is quite frequent to find in Cuenca, and also we can find forest of holm oaks, which are the Quercus ilex, and the Mediterranean maridimum pine, the Pinus pinaster. If you see in this table, the study areas are located in an altitude range that varies between 900 and 1,400 meters. We have found two main soil types, which are the infectisols and the entisols, and the shrub species that appears with the forest are mainly the same, but with some slight changes in the species depending of the place that we have found them. And how have we done the soil sampling? Well, we took 3 plots in every sampling site. In a different, yes? Okay, I'll follow. Can continue. With a difference between plots of 100 meters or so. Then we took 1 sample in every plot, and every sample is made of 3 subsamples. So at the end, we are working with 168 samples. We made 8 samplings in 2011 and 2012, 2 times in every season. And these are the variables that we have been analyzing in this research: the soil moisture, the electrical conductivity, the pH, the carbonates, the total organic carbon, the total nitrogen, the phosphorus content for the part of the physicochemical properties, the basal respiration, the microbial biomass carbon, the dehydrogenase activity, the alkaline phosphatase, the urease activity, and the beta-glucosidase from the biological properties. In this slide, you can see a small scheme of the procedure that we have followed. First, we start with our set of variables in which we make a selection using a principal component analysis. And we choose the highest values that we have obtained. So then we make a correlation analysis once or twice, and then we've got at the end our selected indicators. These indicators that were selected were transformed using a function that can be of 2 types: more is better, or the Gaussian type curve. Depending on the variable, we choose one or we choose another. At last, we've got our standardized variables. Then we make a ponderation and summation to obtain the soil quality index. This procedure, you can see here in a more numerically speaking, we start from 13 edaphic variables, 7 of which were psychochemicals, 4 of them were enzymatic activities, and 2 of them were microbiological. When we make the first principal component analysis, we obtain the table that you can see at the right. And I have highlighted in yellow the result of the highest values that we obtain. You can see here that we obtain a high value for moisture, for pH, for some enzymes, etc. But we make a second principal component analysis to reduce these 8 variables that we obtain up to 6, just to 6 variables that we selected, that at the end were the biomass carbon, the alkaline phosphatase, the beta-glucosidase, the moisture, the pH, and the total organic carbon. We use some standardized function to have all of these variables values standardized. And the final result, well, not the final, but one of the last results are these equations, these standardization equations that we obtain for all our 6 variables. You can see that for the moisture and the pH, we have one type of equation, an exponential type, and for the rest of them, we've got a Gaussian type curve. You can also see in this table some other data like the standard deviation or the slope of the equation. And here is our final result. Here is the final algorithm. And we can say that this index, as you can see, include information about nutrients, microbiological and enzymatical activity, and chemical stability of soils. Also containing all the variables that Zornoza, which is who, if you remember, I have mentioned at the beginning of the presentation, proposed for forest soils. It is also easy to use because it includes just the parameters needed for the analysis, the 6 parameters. All the parameters are also in pairs, so you have the moisture and the pH in one group, the 2 enzymes in another group, and the other 2 ones in another group. So when you want to use it, you have only to introduce the value in the highlighted parts, and you get the result. It is formed by parameters of high sensibility to environmental stress, as the microbial part, and using this index allows us to establish up to 4 quality levels. Those quality levels are this one, a low level, a medium one, a high, and a very high level. Do you remember that I said before that we've got 5 study areas but 7 sampling sites? Well, that was because in 2 of these sampling sites, we have used or we have found altered soils, and we have used them to prove or to test this algorithm, and that's the reason why maybe some of you have seen that the values of the range are a bit low. Actually, in our investigation, we are trying to improve this algorithm to make it even more general, using more data. Here we have used five places. Now we are using 15, trying to, as I said, to improve the algorithm to use it in more kinds and in more type of soils. So what are our main conclusions when we finished this investigation? The first one is that we have achieved to create a valid model to establish the level of functionality in Cuenca's mountain range soils. The second one is that this index includes information of every group of variables that I have been talking before, includes biomass carbon, enzymes activity, moisture, etc. And also allows us to establish different levels of soils quality, low, medium, high, and very high. Also, because of the strong sensitivity of the variables included in the soil quality index, this index is a great tool to use in premature detection of changes in the soil, and also to quantify the impact suffered. So we can say that we have created a great tool to manage forests efficiently in Mediterranean mountain climates, and even now we are trying to improve it to get even a better result. So that's it. Thank you for your attention. I hope you like it. And now, if you have any question, I'll be at your disposal to answer it the best I can. Thank you very much.
Thank you very much, Dr. Francisco. We apologize for the interruption caused during your presentation. Okay, now the session is for open questions.
Can I can I ask one detail?
Yes, yes. Go ahead, doctor.
Okay, thank you. Well, I liked the presentation and I congratulate the speaker. I would like to have some more detail on the way you define the 4 ranks, the 4 intervals for the index. Is that can you show the, yeah, if you can show the last
slide. You want to see the slide of the range, no? This one.
Exactly. Here in the range you have 0 to less than 20, let's say 0.20, then from 0.20 to 0.27, 27, 34. How these borders were defined, these intervals. I did not quite understand that in comparison with the uncertainty of these values.
Yeah, okay. Those ranges are made up because, as I said, we have used the 2 places. Remember that I said that we use 5 areas in which we sample, and there are 2 more. These 2 more places are altered soils, so we have used them to try the final algorithm to use it as a test. Those 2 places. There was a holm oak forest and a pine forest. So we use it as a test to validate the expressions, and the numbers that you see here are the result of this test. Okay, because they are altered, the range is a bit low. Okay, so we know that, and that's one of the reasons that we are trying to improve the expression to have the values of the range in to be not so low. I don't know if I'm explaining well.
Okay, I understand. Okay. Thank you.
Thank you.
Okay, any more questions for Dr.